It is a really good opportunity, especially for all of us with i3L students coming from different background, from different study programs. It's a good thing that you will be able to understand about the topic, which is going to be mentioned by Sir Wisnu, by Pak Wisnu Wicaksono, in terms of the relationship between microbiome, microorganisms, and the environment, and how it interacts or it affects our body, it affects human body. Okay, next book. So before we begin with our talk with Pak Wisnu, next, Ms. Afina, I would like to first of all remind you about the code of conducts. First of all, please kindly fill the Google form, which has already been provided by Bu Afina in the chat box. You can just click on that one and then start filling it up. It's a compulsory one for you to fill it up if you require an e-certificate to be sent to you guys later on. So make sure that you have your complete name over there, as well as your email address, especially the active email address for you to be filled into this Google form. Second one, you have to make sure that you mute yourself during the talk. But feel free to always use the chat box. Yeah, if you have any questions, and then right away, then you cannot wait until the Q&A. Type it right away on the chat box. Feel free to use it so that everyone will be able to just learn, and then Pak Wisnu will be able to tell right away the answer for whatever questions that you have. And then evaluation survey will be shared to you guys later on at the end of the talk. So if you can stay a little bit, and then you can share your opinion regarding the power talk that we have already got, then it's going to be great as well. Next slide. So this is basically the overall general outline of the talk today. We are going to have an opening by Ibu Afina. It has already been done. And then an introduction by me as the moderator today. And then the main talk by Pak Wisnu. It's roughly going to be around 45 minutes or so. And then after that, we will follow by it will be followed by the Q&A session. But feel free to always write down or type down your questions right away onto the chat box. And then later on, I can always read your questions on the chat box, or you can also raise your hands, use the raise hand feature here. No, there's none like that, but you can just unmute yourself and then ask questions right away during the Q&A. And finally, we are going to have a closing at the end of the talk, of course. Next slide. So it's an honor for me to introduce Wisnu Adi Wicaksono, SSI PhD. So Dr. Wisnu, he is a senior scientist from Gras University of Technology. Welcome, Wisnu. With his education background, he actually graduated from Atmajaya Catholic University for his bachelor degree in biotechnology. And then after that, he continued his degree right away into Lincoln University in New Zealand. It's from 2013, where he was awarded Doctor of Philosophy in Plant Microbiology. At the moment, his research focus is in plant microbiome, where he is trying to understand about the indoor plant-associated microbial communities, and then human microbiome, where he is trying to research on the role of environmental microbiota in the development of immune-mediated diseases. And then he's always, when he's trying to understand everything here, he's trying to combine the multiomics approach. So multiomics, we have the metagenomics, we have the meta-transcriptomics, as well as the metaproteomics in order to understand the microbiome communities analysis. And Dr. Wicaksono has been awarded several grants, as well as the award, as you can see from the list here. In order to understand the compositional variation in the bacterial communities of tempe. So I believe this is something that I think Pak Wisnu he's undergoing project at the moment, which is that's part of the reason why he visits Indonesia at the moment, coming all the way from Gras University to Indonesia. And then he was also awarded David Jackson Prize Award for the best research article, and then International Congress for Plant Pathology travel grant. So I believe that his contribution to science as a senior scientist in Gras University of Technology, as well as the another Indonesian scientist who has already gone abroad to get more into the science community, it's going to be a really good thing for you guys to learn from him today. So I believe that's all the introduction from me, Pak Wisnu. If you have anything else to add later on, you can feel free to add. So without any further ado, then let us welcome Pak Wisnu for his talk.
Okay, let me. So is my voice clear, everyone?
Yes.
Yes. So let me share my screen. So can you see the full screen now?
Yes, it is visible now.
Yeah, okay. Okay, so I will start now. So thank you, everyone. Welcome to my talk. It's a pleasure for me to giving my talk today with the title of Linking Plant Microbiome Management with Gut Microbiome and Human Health. So before going to the scientific part, I just want to share with you Graz. So I'm coming from Graz, from Graz University of Technology in Austria. So for those of you that not familiar with Graz, is actually the second largest city in Austria, right after Vienna. It's not as big as Vienna, but it's quite nice living there. So you can see here, this is a snapshot how the city looks like. So one of the nice things, we are surrounded by the hills. So many activities you can do some hiking, go to the forest, get exposed with environmental microbiota that I believe it has a beneficial effect for us. And also, as you can see here from the pictures, the city itself split by the river. So there are two parts of the city between the rivers, which is quite nice. There is a lot of activities that can be done in the side of the river. People do surfing and also walking and running. It's quite nice, especially during the summer. And you also can see here, this is one of the most popular sightseeing if you going to Graz. This is actually a castle, so it's a stronghold that built during a long time ago, and it's one of the most popular sightseeing for tourists. This is another angle. So if you go up to the hill, you can see there is a clock tower that quite famous, where you also can see the overall view of Graz from the hill. So if you're visiting Graz, so you're more than welcome to visit our institute, and yeah. So I will start with the research part. Our Institute of Environmental Biotechnology, we focusing on environmental samples. We try to understand the role of environmental microbiota using a multi-omic approach. So we combine different strategies to understand what kind of microbe in the environmental samples, and also what can they do. So we also investigate the gene profiles using different multi-omic approach. We combine amplicon sequencing, where we try to understand the microbial community composition based on the marker genes. We also functions of the microbe in your particular samples. You also can do metagenomic, where you sequence the entire gene of not only a microbe, but anything in your samples. We also have a facility of microscopy analysis if you want to visualize the colonization of a bacteria or fungi from the environmental samples, from root, from the soil. We also have this a facility to visualize the colonization. And if you look at on the title itself, my talk is about gut microbiome. So you've seen there are words which is a microbiome, which is gonna be the focus of my talk today. And just to give a brief introduction, what is microbiome? So in 2020, our group redefined what is the microbiome. So microbiome is originated from microbiota. So you might have heard this word. So it's composed of any small living organism. It can be bacteria, archaea, fungi, protists, and algae. And also they are theater of the activity. So not only the cells, but also what they can do. So it can be their protein, lipid, carbohydrate, their nucleic acid, also their mobile genetic element, so including virus and phages. So to differentiate the microbiota and microbiome, for instance, virus is not microbiota, but it's a part of microbiome because it's part of the theater of the activity. So microbiome normally occur in the specific biome. So depending on what kind of samples, you might have seen different terminology. For instance, soil microbiome, which is the biome is soil. So any microbiota with the theater of the activity that occur in the soil. And also there is other terminology, for instance, resistome. If you break down the word itself, it's coming from the antibiotic resistant gene. So all of the antibiotic resistant determinant within the microbiome. That's why we call it a resistome. And why microbiome is such a hot topic currently, not only in Europe, but also around the world, because they play such an important role. This is one of the examples of what kind of roles that microbe play for human. They help us to digest the protein, also train our immune system, but just look at the number of microbes across of the human body. So actually we are more microbe than human, because the number of bacterial cells actually overcome the number of human cells in our body. So if you look at the pictures here, there are roughly 10 until 100 trillion of bacterial cells occupy our body. And you might heard about the gut microbiome, because this is one of the part of microbe that plays such an important role for human health. Many diseases are associated with the gut microbiota composition. So for instance, if there is a dysbiosis or loss of diversity in our gut microbiota, we might get some diseases. For instance, asthma and diabetes, they are associated with the reduction of bacterial diversity in our gut. And also, if you look at the number of bacterial genes across in our body, it's 100 times more than the number of human genes. So indeed, the human cells play their own functions, but also there are microbes that play important roles in our body. Like I mentioned, they help us to digest a protein and also produce a secondary metabolites, protect us from the pathogen, and etc. So microbe is important for the host. And not only in the human, but practically microbe can be found everywhere, even in the deep sea, where there is only a small number of living organisms that can live in the deep sea. Microbe play important roles in the terrestrial ecosystem. So you might have heard about rhizobia, which is a type of bacteria that can fix nitrogen. So it helps plants to uptake nitrogen from the soils. So this bacterium can fix the nitrogen from the air and make it available for the plants. And also you might have heard about arbuscular mycorrhiza. So this is a typical fungus that colonize inside of the root tissue of 80% of most terrestrial plants. So many plants that grow on the soils, they have a arbuscular mycorrhiza colonization, which is important to provide nutrient, for instance, phosphate for the plants. And also many other functions that microbes provide to support ecosystem functioning, photosynthesis, mobilization mineral, not only in the terrestrial ecosystem, but also in the aquatic ecosystem. So there is a lot of biogeochemical pathway that are played by microbes. So that's why microbe it's important to support the ecosystem functioning, and any factors that might disturb microbial commodity composition, it has a substantial impact of the ecosystem productivity. I mentioned to you before the importance of microbes, but I will focus on plants and also and human, but I will start with the plants. So we call it as a holobiome. So it means the living organism, including the microbe that living inside of the host. So that's why we call it holobiome. And the microbe play an important role to define the health status of the host. Because, for instance, different phenomena can be defined by the microbe. For instance, microbe can help plants to germinate and also protect plants from the disease. We do have a research where we found one bacterium called Stringomonas in rice. So without this bacterium living inside of the rice can get a disease that's caused by other bacterium called Burkholderia. So the interaction between the antagonistic bacteria and also the pathogen is important to define the health status of the host. So if the bacteria that play an important role to protect the plant from the disease decrease in the relative abundance, the plant to tend to have the disease that's caused by the pathogen. And one of the things that I would like to highlight is the microbial diversity. So it's the number of bacterial species or fungal species or microbial species in the samples, so in the plants. So this microbial diversity defines the health status of the host. So in many cases, what we observe from our study, if living organism, plants, human, they have a disease, they tend to have less number of microbiota living inside of their tissue, because there is a disturbance in the microbial community that can reduce the diversity of the microbiota. So biodiversity is currently used as a biomarker to explain changes in the microbiome. And one of the things that quite interesting, actually, the plant microbiome it's unique and specific. So if you sequence, investigate the microbial community composition from different plants, for instance, rice, and you compare with tomato, you might observe different microbial community composition, which means the plant genotype community composition. So plant can attract specific microbes, so they can colonize the host and also provide a beneficial function for each plant. And we have performed such a study to explain if plant carry a unique bacterial community. So we close to the Alpine mountain. So this is an ecosystem where has been established for 1,000 years without any disturbance from anthropogenic activities. We went to the Alpine and also collected a sample from different native Alpine plants. So we collected the seeds from the different plants that grow in the same soil. So we go to one spot in the Alpine, and in this particular spot, there are multiple different plant species that grow together. So you might expect they carry the same microbe because they grow in the same soils which have similar microbe. But actually what we observe is different plants, they carry their own microbial community. So this is different seed of plants that we collected, and we also combine it with microscopy analysis, so we can attack the microbe inside of the plant tissues by bind them with a specific group. So for instance, if you're interested with gamma proteobacteria taxa, you can design probe that only can bound with gamma proteobacteria, and it reflects a fluorescence where you can see the colonization of bacteria using a fluorescent microscopy. So this small dots, red dots here, it's a colonization of bacteria inside of the seed of different plants. So in this study, we perform amplicon sequencing to investigate the microbial community composition. So you can perform a PCR on the marker genes, so you can identify what kind of bacteria that present in your samples. And from different plant tissues, I think we analyzed around 20 different plant species that grow in the same soil, so in the same plot. When we combine all of the data set, we observed there were almost 12,000 of bacterial OTUs. So OTUs is operational taxonomy unit, so it's equivalent with bacterial species. So we observed there is almost 12,000 bacterial species in the whole plant species, but only 11 bacterial species that share from different plants that grow in the same soils. So plants, they have a specific microbial community composition. They attract specific microbes that's not the same with the other plants, because each microbe, they have their own function, and plant also need specific function from the microbes. So that's why they have a different bacterial community composition. We also observe the same pattern from the fungi. There are almost 4,000 fungal species that we found from the entire data set, but only 5 fungal species that share across different plant species. And this is another picture after we germinate the seed. So right after the seed was germinated, the root tissue was immediately colonized by different bacteria. So the bacteria comes from the seed, and by the time the plant develop, they disperse to different plant tissues, including the root tissues. So plant microbiome is unique. So different plant species have their own microbial community, which is important to support the plant development. And as I mentioned, microbes are everywhere, and my study especially focusing on the impact of the anthropogenic activities on the loss of biodiversity in our fresh produce. So in the next few slides, I will focus more about plant microbiome, but mostly from the fresh produce, so either fruit and vegetables. And why this is important, because we consume fruit and vegetables, but they also carry their own microbes. So any anthropogenic activities, intensive agriculture, antibiotics, pesticides, breeding, they might have an impact on the indigenous plant microbiota from our fresh produce, and eventually might influence the gut microbiota, because we consume potentially we consume the microbes that occupy our fresh produce. And this is just a brief introduction about the Anthropocene. We're facing rapid human population growth, also urbanization, over consumption, and also intensive agriculture, and all of these factors directly or also indirectly impact the plant microbiota composition. And we also already crossed several boundaries here. You might have heard about the biodiversity crisis. We lost a lot of animal diversity and also plants, but one of the factors that normally overlooked is the diversity of the microbes, because climate change, for instance, also decreased the number of microbes in the soils, and it has been proved from multiple studies. Using pesticides, it decreased the number of arbuscular mycorrhiza species in the agricultural system. And this is important because I mentioned to you before, microbes play important role to support the ecosystem functioning. Pollution and microplastic is also one of the key challenges that we need to face in the next few years. Nitrogen crisis and also antibiotic crisis. In 2050, it was predicted that antibiotic-resistant bacteria can be the major threat for a human. We running out the number of clinical antibiotics that can treat emerging pathogens. So we observe many emerging pathogens now can infect human, but we cannot treat them with our clinical antibiotics. So we're running out a solution to eliminate the opportunistic pathogen. And one of the focus that I want to bring up is what is the impact of the Anthropocene, so intensive agriculture, human population, how they have a direct impact on the plant microbiome from the field, and also to the fork, which is something that we consume from the fresh produce point of view. So in my presentation, I will share my study from our group. I will split them to 3 different parts. So started from the impact of the anthropogenetic activities from the field, so how they change the microbial composition in the field, and also fresh produce. Normally after harvested, they undergo a post-harvest treatment, and this is one of the major factors as well that influence the microbial composition, and how these 2 factors is connected to our gut microbiome and how they can define our health status. So this is another introduction about the importance of the food microbiome. I think it's important to bring this up because food microbiome play an important role on the development of our gut microbiota. So this is a picture of different factors that influence the gut microbiota development. So in this study, it's part of one of the most comprehensive studies to investigate the development of gut microbiota from the early life until approximately 4 years old. So in this study, they collected the stool samples from a baby that have around 3 months old. So they collected in the stool sample in monthly basis until 40 months old. So they perform a microbial community analysis using a metagenome sequencing, and they combine it with such a rich metadata. So every cohort that was analyzed, they do information about the birth mode, whether it's a C-section or normal delivery, the gender, the breast milk, and also when the solid food is introduced, consumption of probiotic, different geographic locations, and the number of siblings in the same household. And I just want to point out the major factors here. So the longer the bar, indicating the higher impact on the gut microbiota development. Indeed, the breast milk, so the consumption of whether the baby consume breast or also formula milk, is one of the major factors that influence the gut microbiota development during the early life. But also one of the things that I want to point out is the solid food. So normally during the early life, baby consume fruit and vegetables that has been processed, for instance, puree or boil, and the consumption of solid food and when it was introduced play an important role on the gut microbiota development. And why gut microbiota development is important? Because they are associated in the development of many diseases. Because in this particular period between 1 until 2 years, that's the time when our immune system was trained, not only by our human body, but also from the environmental microbiota. So the diversity of the gut microbiota and also the exposure to environmental microbiota, it's important to train our immune system. Many diseases are associated with the lack of development of our gut microbiota, asthma, celiac disease, and other immune-mediated diseases. They have a high association with the lack development of gut microbiota during the early life. So one of the highlights of this picture is the solid food has a crucial impact on the gut microbiota composition. And as I mentioned, that typical food, solid food that consumed by the fruit and vegetables. So this typical solid food is important because we hypothesize that the intake of plant-associated microbes is the main source of the human microbiome to the environmental microbiome. So when we consume fruit and vegetables, because they carry their own microbe, there is chances where the microbe inside of the fruit and vegetables can be transferred to our human gut and contribute to the overall gut microbiota diversity. And this is another microscopic colonization from fresh produce that we normally consume. Here we have a research on lettuce, where we observe under the fluorescent microscopy different bacterial colonization. It might be slightly difficult to be seen here, but you can see here there is a lot of red dots. We tag the bacteria with specific probes, so we can visualize the bacterial colonization inside of the lettuce in the part where we normally eat. This is arugula, so the green color here indicates the bacterial colonization in arugula, and this is apple. So you can see here multiple yellow dots here. So there are bacterial colonization inside of the flesh of apple. So the parts of fruits that we normally consume, there are living bacteria that live there. So all of food plants, fruit and vegetables, they are associated with high microbial diversity. There are a lot of different bacterial species that colonize different plant tissues and different type of plants. So just to give you a perspective, this is also from our study. We use a qPCR to calculate the number of bacterial cells inside of the plant. So if you eat one apple, you probably consume 10 to the power of 8 bacterial cells from the whole apple. And also there is approximately 1,800 bacterial species in the whole apple. So when you eat the apple, you don't just eat the apple or consume the nutrient, but there is possibility you also intake the bacterial cells because they are living in the apple tissue. So our main hypothesis is to connect between the microbiome from the field and also the gut microbiome, because we consume the fresh produce. So any factors that influence the indigenous fruit microbiota composition that might change the composition might eventually impact the gut microbiota composition. So I will start to share our study. So I start from the field. I mentioned to you before that there are multiple factors, anthropogenetic activities that might influence the microbial commodity composition of our fresh produce. Plant species, definitely one of the major factors, but also soil type. So different soil, they carry their own microbes. So if you group the same plants in two different soils, they may also carry different bacterial commodity composition. And also agricultural practices, we use a lot of pesticide, fertilizer. It puts such a huge pressure for the indigenous microbiota, which eventually change the microbial commodity composition. But in this talk, I will focus on the plant breeding. So you aware that the plant breeding is normally intended to generate a plant with a specific phenotype, increase the productivity, and also if you want to generate the fruit with specific color or specific size, that's one of the main goals for doing the plant breeding. But also it's not only change the plant phenotype, but also the microbe inside of the fruit itself. So we have a study that published in 2016, and also we recently published in 2021. So we used the Styrian pumpkins and as our model. So in Austria, we breed different pumpkin to generate a specific seed that we want, because we collect the oil from the seed of the pumpkin. So we do have different inbreed lines. So let's call line A until D. So we cross different lines, as you can see here. So the line A until line B, they practically have a similar shape and color of the seed. So if we combine line A and line B, we get this cultivar core diamond, which is slightly different compared than their parents. But not only the differences in the phenotype, but also like a human, when we breed two different cultivar, the lineage of the two cultivar, they carry microbe as a part of their parents. So when you combine line A and line B, the cultivar diamond, they have a fraction of microbe that originated from line A and also from line B. So the plant breeding not only changed the phenotype, but also changed the microbial composition. And many studies show as well that, for instance, if you compare the wild type cultivar with the commercial cultivar, you might also observe the same thing. So there is a changes in microbial composition. And also what we observe from our study is there is a reduction of microbial diversity from the commercial cultivar. So the commercial cultivar, which is intended to increase the plant productivity, but when you analyze the microbe, so they have less microbial diversity compared than the wild type cultivar. So the plant breeding not only change the microbial composition, but also tend to decrease the microbial diversity in our fresh produce. And this is another study that we still under review. So if you ever wonder the microbial composition from the fruits that you buy from the supermarket, whether it's different from those that are obtained from the private setting, from forest, or also from the home garden. So this is what we did in this research. So in Austria, especially in the case of apple and blueberry, people tend to grow their own apple in their garden. So if you go to Graz, you might have seen in front of someone's front yard, you might have seen the apple tree, because they tend to grow their own apple, and they consume the apple that grow in front of their houses. And also the other way, obviously, to obtain the fresh produce is by purchasing them from the local supermarket. And we were wondering if they do have a different bacterial composition, because we consume this food, so we consume the fruit and vegetables that we obtain from the supermarket or also from our local garden. So we perform amplicon sequencing. So this is approach to investigate what kind of bacteria or fungi in the samples. So we collected apples and blueberry from the supermarket, and we compare them with those that grown from in the natural setting and also in private houses. And what we observe from this study, so this is an NDS plot. So every dot indicating one sample. So if one sample is close to each other, so it means they have similar microbiota composition. So here, for instance, the green color, this is the apple that we bought from the local supermarket, and the red color is apple that we picked from someone home garden and also from the natural setting. You see here the green color, they tend to cluster together. So we bought the apple from different supermarkets, and also they do have different cultivar, but still they cluster together. So it means that the fruit and vegetables, in this case is apple, that we normally purchase from the supermarket, they have a similar microbiota composition. So the intensive agriculture tend to shape the microbiota composition in our fresh produce more similar. And from those that are obtained from the local garden, also from natural setting, that tend to spread. So it means they have a completely different microbial community composition among each other. And why this is important, because I mentioned to you before that the microbial diversity, the high microbial diversity is important for the host. So if we consume apple from the local garden or also from the natural setting, we get exposed with more diverse bacterial community, which is important for our health. We also perform another analysis to investigate the signature taxa from different foods that we obtain from the local supermarket and also from the natural setting. I just want to highlight the Pseudomonas and Ralstonia and also Snetophomonas. So these two different bacterial genus have a high abundance in the foods that we bought from the local supermarket. And why this is important, Ralstonia, normally known as the emerging pathogen, it also has a synergism with the E. coli, another emerging pathogen. So this typical bacterial genus is known to have a harmful effect to human, and also Snetophomonas. So we also perform another sequencing to identify the species name. So it belongs to Snetophomonas maltophilia, and this bacterium is also have a high abundance in the fruits that we obtain from the supermarket. And maltophilia is known as opportunistic pathogen that can make cause a disease in humans. So the intensive agriculture from the horticulture foods that tend to increase putative taxa that have harmful effect for humans. In contrast, those foods that are obtained from the natural setting and also from the local garden that tend to have more beneficial taxa, for instance, Pantoiae, you can find it here. It's one of the known biological control that can antagonize Erwinia, which is another pathogen in apple. We also observe there is a high abundance of Enterobacter hormaici, is one of the bacterium that can increase yield and protect plants from the disease in tomato. So there are more beneficial taxa from the fruits that grown in the natural setting. So now I'm entering my second part, managing the post-harvest microbiome. By the time the fresh produce are harvested, they normally undergo the post-harvest, so it can be treated with pesticide. For instance, in the apple case, in the United States, they normally wax the apple, so it makes apple more shiny. So if you go to the supermarket, you might wonder why apple is always looks shiny, because they put a wax on the skin, and it's actually also changed the microbiota composition. We discussed this in review in FAMS, so you might check this. We propose to use more microbial inoculant as an alternative strategy to replace the synthetic pesticide. We also propose to use a chemical booster to increase the efficacy of microbial inoculant, so our fresh produce is more environmental friendly. And this is one of the studies that I would like to share about the impact of the post-harvest. This is a Dr. Wasserman, part of our institute. So she was interested to investigate the impact of the global market, in this case is apple, on the microbial community composition, but also the resistance, so the antibiotic resistance inside of the apple. So what she did was she went to South Africa. So South Africa is known as a country that produces apple. And normally apple from South Africa, they are shipped to Austria. So it might take one or few weeks to reach Austria. So the apple undergo the post-harvest treatment. They are stored, they are shipped until it reaches the local supermarket in Austria. So what she did, she performed a microbial community analysis using a metagenomic approach, not only analyzing what kind of microbe in the samples, but also their gene profiles. And what she observed from the two different apples, so she collected two different apple cultivar from South Africa. So she took a fresh apple from from the field, and the same cultivar that has been shipped to Austria. We compare these two cultivar, which called Braeburn and Royal Gala. This is two of the most famous consumed apple cultivar. And she observed a shift in microbial commodity composition from Stromodales to Enterobacteriales. So Stromodales is normally known as a plant growth promotion bacteria. So it helps plant to produce growth hormone and also protect plant from the pathogen. And just by storing and ship apple to different country, there is a shift from the good bacteria to Enterobacteriales. So Enterobacter is known as a group of bacteria that cause a disease in human. You might heard about the E. coli, Salmonella, and Shigella. This is part of Enterobacteriales. And not only the microbial commodity composition shifted to the composition that more harmful, but also there is a changes in antibiotic resistant gene profiles, because the antibiotic resistant, they are carried by the microbes. So if there is a changes in the microbe, you might expect there is a changes in antibiotic resistant genes as well. And what we observe, because Enterobacter is normally opportunistic pathogen, they have specific antibiotic resistant genes. We also observe there is a storage specific resistome. So from those apple that has been stored and shipped to Austria, we found more clinical antibiotic resistant genes and also more plasmid encoded antibiotic resistant genes. And this is not good, because this antibiotic resistant gene can be horizontally transferred to the opportunistic pathogen and cause a disease in human. And this is another factors that I would like to discuss is a cooking process. So it just recently published last week. So in this study, we try to understand if the cooking practice might also change the microbial commodity composition from our fresh produce. So again, we use apple. And why the cooking practice is important, because during the early life, normally infants consume different type of fruit and vegetables. Normally it's boil, they puree, they spread it. And we were trying to understand if this common practice to prepare food for the babies, it might change the microbial commodity composition in our fresh produce. So before going to the impact on the microbial composition, this is pictures of different bacterial colonization inside of the apple flesh. So you can see there are multiple color here, because we tag the with different probes. So the yellow one is indicating the gamma proteobacteria, and also the purple one indicating the beta proteobacteria. And inside of the apple, like I mentioned, it has high diversity of bacterial species. And one of the key highlights from this study is we also cultivate the bacterial isolates from the apple. And a lot of them actually have beneficial properties. For instance, we challenge them with opportunistic pathogen. In this case, it's Acinetobacter, E. coli, and also Pseudomonas aeruginosa. It's typical bacteria that cause the disease in human. And those bacteria that are isolated from the apple flesh, they can antagonize this opportunistic pathogen. And also the high proportion of bacterial isolate can also produce a protease, which is important for our health. And also they are resistant against bile salt. And this feature is important because normally bacteria that resistant against bile salt, they might survive in our gut system. So to investigate the impact of the cooking practice, we treat apple differently. So we compare raw apple with shredded apple. So shredding apple is a common practice in Austria, where the baby consume shredded apple because it's more fine, the type of the apple. And also we boil them, make a puree, pasteurize the apple, and also dry the apple for 6 hours to make it a dried apple. So this is typical snack for children. And one of the highlights of this study is even though we boil or dry the apple for a long time, there is still bacteria that live inside of the apple. So this is from another microscopy analysis to prove that there is living bacteria in the apple. We use a live and dead probe, so we can differentiate if the bacteria is still intact or they are not intact. So this dye can bind with living bacterial cells. So those that are living are colored by green. So you can see here there is a lot of green color here, which indicating intact bacterial cells, but also there is a lot of dead bacterial cells here in the raw apple. And after we boil the apple, you still can see the green dot color, so indicating there is still living bacteria even after we boil the apple. And also after we dry the apple for 6 hours at 60 degrees Celsius, we still see some bacterial colonization inside of the apple. So our common cooking practice, they didn't eliminate completely the microbiota inside of the apple. And also the composition changed. So obviously we observe the reduction of bacterial diversity because the heat treatment kill many bacterial species. So we observe the reduction of microbial diversity, but also the composition is changed. So it's not similar anymore with the raw apple. And why this is important? Because in raw apple, we actually observe a lot of bacteria that have beneficial function. So our cooking practice have an impact that might eliminate a beneficial bacteria in the apple. So as a summary from the first 2 parts, we've seen that the plant microbiome already changed from our anthropogenic activities. We observe a decrease in diversity and also the evenness. We observe there is a changes in microbiological composition to more that tend to have a harmful effect. And also we observe there is an increase of antibiotic resistance from our fresh produce. And to link those two is we have to prove that if the bacteria from our fresh produce can actually transfer to our gut and whether they contribute to overall gut microbial diversity. So we know the anthropogenic activities change the composition from our fresh produce, the microbial composition from our fresh produce, but is it true that the microbe from the fresh produce can colonize our gut system? So to prove this hypothesis is we perform a meta-analysis. So we collected fruit and vegetables metagenome. So if you're familiar with multi-omic approach, if you have a metagenome samples, you can construct a bacterial genome using a particular bioinformatic approach. So we collected around 156 metagenome samples from the database, and we construct bacterial genomes. We obtain roughly almost 1,000 bacterial genomes from different fruit and vegetables metagenome. And to prove that if this bacterial genome present in our gut, we also combine our genome catalog. So we have a genome catalog that are originated from fruit and vegetables. And also in the database, there is a genome catalog that the bacterial genomes are originated from the gut. So if we combine these 2 genome catalog, we can differentiate if we find this bacteria, whether it's originated from the fruit or also it's just the indigenous gut microbiota. So we combine these 2 data sets and obtain also the gut metagenome from the database. So we run approximately 3,000 gut metagenome to see if the bacteria that living in our gut, whether they are originated from the fruit and vegetables or is just the indigenous gut microbiota. So this graph is just to show the different bacterial taxa that are originated from the human gut. So mostly this is indicated by the red color. So it's those bacteria that are originated from the human gut. So mostly they are belong to bacilli and also Clostridia. But those that are originated from the fruit and vegetables that are colored by blue color here, they are mostly gamma proteobacteria, alpha proteobacteria, and actinobacteria. So plant and gut, they do have different bacterial community composition. So we map the metagenome reads to identify the fractions that are originated from fruit and vegetables, and also those that are originated from the gut. And what we found is from, in average, in our gut. So for instance, if we have bacterial diversity there, 2.2% of the total bacterial diversity are predicted to be originated from fruit and vegetables. So if we consume a fruit and vegetables, there is a potential that those bacteria that living inside of the fruit and vegetables, they can survive in the human gut. And also there are multiple factors that influence the bacterial diversity in the human gut. So for instance, we collected the data from American Gut Project. So they collected the stool metagenome from more than 2,000 cohort. And what we observed, so we mapped back again our genome catalog and found that if we eat vegetables in daily basis or regularly in your gut, we can find more bacteria that are originated from fruit and vegetables. And also, if we consume a different type of fruit and vegetables, we also observe a higher bacterial diversity in our human gut. And why this is important? Because again, I mentioned that the higher bacterial diversity is important for defining our health status. So if we get exposed by more diverse bacteria by eating more fruit and vegetables, it increases the bacterial diversity in our human gut. And why this is important, we currently facing the biodiversity loss. People tend to live in the urban area. They get less exposed with the environmental microbiota, and all of this are associated with the development of immune-mediated diseases. And this is one of the studies that prove that the exposure to high diversity of microbiota can regulate our immune system. So in this study, the author do intervention treatment. So this is, they collected the stool sample and skin samples from kids that go to daycare. So the playground of in the daycare, it looks like this. So it doesn't really have a natural setting. So they collected the stool and skin samples, and also they checked the immune protein from those kids. And after that one, they convert the playground to become more natural setting, and also they collected the same samples. And what they observe is they observe a higher microbial diversity in the skin and also stool sample from the kids that the playground has been changed to more a natural setting before and after. And this is the response is actually similar with, so they also compare with the kids that go to the daycare that more natural best. So it's almost similar with those kids, because those kids also normally go to the forest, and they also have a higher microbial diversity in their skin and also stool sample. And this is related with the increase of immune-related protein. So by exposing us with the high microbial diversity, it enhances our immune-related protein. So this is the last slide from me today. I just want to propose, this is the vision from our institute, using more probiotic to manage the plant microbiome, because it's important to eventually define our gut microbiota development and our health status. And we have a platform that also I would like to share. We have a open course that if you are interested, just check this link, and or also you can scan here. You can also register. We do have multiple courses. It's designed not only for scientists, but also for general public. In this course, it contains information about the importance of microbe in different aspects of our life. And also we actually design games for doing like microbial games, so to make it more interesting. So it's also, I found it is quite refreshing. So yeah, this is just concluding remark. I think this is quite relevant in our situation now. During the pandemic, we use a lot of disinfectant. We try to eliminate the virus, but also just be cautious because it might also eliminate the beneficial bacteria in our surrounding. So I want to thank you for your attention, and I'm looking forward to receive your questions.
Okie dokie. Thank you very much, Pak Wisnu. What an interesting topic that you have already mentioned there in your talk. So we have already got several questions here in the chat box. Let's start with Alvin from biomedicine. So he wants to ask regarding the wax layer on the surface of the apple. Is it true that apple skin along with the wax should be peeled out? Or is there any other advantages if you just have the skin apple? Just will it affect anything in our gut microbiota at all or not?
Okay, yeah. I mean, I think maybe it's also important to peel off the apple because I'm not fully aware if there is a study that investigate the impact of the wax for our health, but it's definitely changed the microbial composition because if there is a wax, it close the pore in the skin of the apple, and eventually it changed the physiology of the apple fruit as well. It changed the microbial composition not only in the skin but also inside of the apple tissues, and we observed this in our other studies. So waxing is normally, as far as I know, it's not applied in Austria, so there is no apple that gets waxed in Austria. Mostly they are more organic treatment in Austria, so there is no pesticide. So with the fresh produce that normally consume, we treat them with less pesticide. But for those that are shipped from the United States, because it's undergo long storing and also ship that takes more time, so they normally wax the apple so it looks more shiny. They even treat with some antibiotics to protect the apple from fungal disease. And this is something that not everyone fully aware, I think, but also it not only have a harmful effect for human, but also it changed the microbial composition. So yeah.
Okay, I'm just wondering, Pat, so relating to the apple skin on your research, mostly you compare, you took the samples from the peeled apple, yeah? Not with
Yep.
the skin. What about do you have any idea about the microbiota in the skin of the apple itself, or is there any?
Yeah, the other factors that actually influence the microbial composition is different plant tissues, because different plant tissues, they secrete a different exudate to attract the bacteria. For instance, if you compare the root samples and the stem samples, they completely have different bacterial composition. It's also occur in the small microhabitats. So if you just analyze the skin of the apple and you compare with the flesh, they do have completely different bacterial composition. Yeah, even in the seed, which is inside of the flesh, we do have this study that published in 2009. You can check I think it's called An Apple a Day. So we Dr. Wasserman split, separate the apple from the skin, from the flesh, is also the upper part of the apple. I don't exactly remember the name, and also the below part, because that's when during the fruit development, that's the two parts that develop first. Then after that one create the flesh, also the seed, and all of them have different microbial composition. And the cultivation practice, either organic or more conventional treatment, they completely change microbiology composition from each plant tissues, even though they are from the same apple cultivar.
Okay, cool. Thank you, Pak. So second question is from Miss Junaida Astina. So it was very interesting. I wonder, how did the bacteria composition change in different cooking process? Was it only due to death of good bacteria, so the proportion of good bad bacteria changed because of the cooking process itself? Or was it due to the growing of new species of bacteria, which was not there before it was cooked, but then after cooking, it starts growing there? So any information from your side, Pak, about that?
Yeah, I think growing, it's because we treated the apple in a short period of time, unless the dried, because it took 6 hours. So the changes mostly because the cooking practice eliminate the dominant bacteria. So those dominant bacteria that live inside of the apple, they are not resistant against heat. So mostly they are eliminated, and which is most of them actually the good bacteria. So those that are survived during the boiling or also long heat treatment, they are more resistant to heat. So the cooking practice actually shape the microbiology composition and filter them to those that more resistant against heat. And unfortunately, those that are resistant against heat, they tend to have a harmful effect. For instance, we observe there is a Pseudomonas and also Ralstonia survive after the cooking practice, because this is typical R-strategist bacteria. By the time the dominant bacteria get reduced, they just proliferate quickly and replace the dominant one. So yeah, I don't think it's because there is a growing of new species, but rather than eliminating the dominant one. So that's why we observe a shift, substantial shift changes after the cooking practice.
Okay, so it's basically the bacteria which were not dominant before cooking, it was already there, but then after the cooking process, that it starts appearing into the test, and then the data is showing there the changing of the composition. Okay, cool. Next question from Alicia Cherry Chandra. So biotechnology cohort 2021. I would like to ask, how does good bacteria change into bacteria that causes disease in fresh apple that are stored and shipped to supermarket? Like it's probably post-harvesting process here.
Mm-hmm. Yeah, I mean, the good bacteria is still there. It's just the composition change. So the post-harvest treatment, normally there are multiple treatment that normally the fresh produce were underwent. For instance, storing in the cold temperature to preserve the fresh produce. In other cases, using a pesticide to treat the fresh produce, and also waxing in the case of apple. And this fresh produce eventually reach the supermarket, and those that are normally purchased by us. And the good bacteria is still there. It's just the commodity shift to, so the dominant bacteria is not only the good bacteria now, but like some of them are harmful bacteria, because this selective pressure actually selects those that are have R strategies. Like I mentioned, Ralstonia, Pseudomonas, they have more resistance towards this post-harvest treatment, and that's the reason why this less beneficial bacteria can proliferate, because this is the environment that they like. So the post-harvest treatment storing, it shifts the composition, and the dominant bacteria is not the normal indigenous microbiota.
Okay, cool. Any other people wants to any other questions from you guys? So thank you from Junaida, thank you from Alicia. They say thank you. Anything else, guys? Do you want to ask? This is the chance for you to really ask questions related to gut microbiota if you're interested in it. So Clarissa, I'm wondering if those microbes that we get from fruits and veggies can survive in our gut, will stay there forever, or they have their own lifespan. So more into how they survive in our GIT. Vishnu, are you still there? Seems like there is. Okay, it seems like there is an internet connection problem with Sir Vishnu, with Pak Vishnu connection. So shall we wait a little bit, Bu Afida? Yeah, let's wait a little bit, guys. Sorry about the internet connection. I believe that. Oh, there you go. You're still muted, Pak. Welcome back, Pak. You're still muted. So, question from Clarissa: can they survive our gut, our GIT, gastrointestinal process with the digestive system, Pak? And then will they stay like forever, or what happened to them?
So can you repeat again the question?
So I'm wondering if those microbes that we get from the fruits and veggies can survive in our gut, will stay there forever, or they have their own lifespan?
There is a data that I didn't share during the presentation. So we also have a longitudinal study. So we collected the data from a published study. So they collected the stool samples from 3 months until 4 years. We also mapped back this genome catalog, and there is a bacteria that still persists until 4 years old, but not all of them. So there is a succession during the early life and change to, for instance, during the early life, there are more gamma proteobacteria associated with fresh produce that more abundant during the early life. But after that one, it's replaced by the lactobacilli or Clostridia, because during this time the gut system, it's more acidic, so it's more selective to those bacteria that can resist against gut acid. So yeah.
Okay, so it means that in our GIT, is it like only the acid from the stomach and the bile salt that kind of like filter the bacteria, or is there anything else in our digestive system that kind of like changing?
Yeah, I think that's the two major factors that filter the bacteria. But I think now is there's the dominant bacteria that normally survive in our guts, mostly Lactobacillus bifidobacterium. They can survive there because it's fits with their requirement to live. But also there is a thing called rare bacteria. So there is bacteria that not dominant, and actually they are important train our immune system. So somehow our mechanism, they also select this rare taxa, which is not typical bacteria that not lactobacillus and also not bifidobacterium, but they play an important role to train our immune system.
Okay, so last question, yeah, for this one, guys. So we get Defina. So I copy pasted again into the chat box, but I believe because you were disconnected, then you could not see it anymore. So yeah, from Defina, she's currently working with food probiotic projects. And then it's assumed that it's probably related to the gut microbiota. So for the project, the bacteria was exposed to heat stress during the processing. So the process of creating the probiotics powder requires them to use heat stress at up to 150 degrees Celsius. Is there any possibility that the bacteria now can develop new properties due to the stress? So can you see the question in the chat box?
Yes. I mean, if I can assume, there is possibility that can develop new properties. For instance, selective pressure, they can make the bacteria mutate, so they change their genotype, and they might develop some new properties. But this is something that you might need to check truly. I think it's not simply just by a few generations the bacteria can have a new properties just by put a selective pressure, but maybe in like 100 generation, they can develop new properties. This is the case for antibiotic resistance. If you treat the bacteria with the same antibiotic again and again and again, and this antibiotic can make a spontaneous mutation that make the bacteria resistant. So they have a mechanism to protect themselves. So bacteria is actually quite smart. So yeah.
Yeah. Okay. So it's probably it's more into the activation of the gene, one particular gene that kind of like can trigger some sort of like changes in the bacteria itself. Okay, cool. Miss Afina, I believe that that's pretty much it, right? So we only have time until 2:30, is it, or 2:15 or something like that?
Yeah. The maximum is until 3:30 West Indonesia time zone. Okay. Thank you, Pak Mario, Pak Wisnu, for the interesting presentation, and thank you everyone for asking questions. Don't forget to fill the attendance too in the link that I just shared again in the chat. Okay. And this is as a form of appreciation, we would like to send you this certificate, Pak Wisnu, signed by our head of department from biotechnology.
Thank you.
Yeah, we really appreciate your time for being the speaker in this month's Power Talk. And also, please, before you leave this Power Talk, kindly fill the survey in this link or in the QR code. You can scan it too. And that's just it. Thank you again, everyone. And
so sorry, Miss Afina. So this is like I think this at the beginning of the Power Talk, probably when the attendance was not here. Pak Wisnu also mentioned something about like his own project related to Tempe. And one of the slides they mentioned about probiotic and stuff like that. Remember that i3L students or students from any other institutes, you have the ability to apply for ISMA later on. So if you're interested in gut microbiota, if you're interested in this kind of like project, like Pak Wisnu is over here already, and then later on, if Pak Wisnu doesn't mind, and probably if someone can approach one of us in i3L asking for your email address or something like that, probably asking for you to become their supervisor later on. Maybe they're planning to apply for ISMA to travel to Graz University of Technology. Then that's probably something that we can also do. Yeah, Pak Wisnu?
Yes, I mean, you can share my email to everyone. So it's not only for the internship, but so if you're willing to collaborate or anything, and also visiting our institute, so you are more than welcome to email me and we can discuss further. So yeah.
Okay, cool. Thank you very much then, everyone. There's a question. Should we all fill the attendance? Not really, yeah? So just...
No,
okay.
The evaluation survey form is different from the attendance one. So in the evaluation form, we would like to know your opinion so we can improve the next Power Talk. That's it.
Okay then. Cool. Thank you very much, Pak Wisnu. Thank you very much, everyone who is attending it. Have a good day, everyone.
Thank you, everyone. Bye-bye.
Bye-bye, Pak Wisnu. Thank you.